For those waiting, we'll be starting in a couple minutes. Suppose I can do that. Maybe get another minute. Looks like there's still some people jumping on. All right, may as well go get started. Hi everybody. I'm Greg Watkins Caldwell. I'm one of the PIs on the ZooMu uh, Research Coordination Network grant, and you're at the April 9th, 2026 Cyber Infrastructure meeting or webinar. Housekeeping, this is being recorded. We're going to share this on the ZooMu website and the ZooMu YouTube channel. So that link will be going out to all the registrants afterwards. The open chat is disabled, so you shouldn't be able to chat through Zoom with all the other participants, but you can with the speakers and the organizers. We're going to be using the Q&A session, and hopefully that works for you. Apparently, there's an issue with which version of Zoom you're using and whether or not you see a Q&A. And if you don't, just put it in chat. We'll see it. The agenda for today is a little introduction about ZooMu, information about the ZooMu workshops, and recent advancements we've done with cyber infrastructure and what we hope our next steps are. All right, so the goals of the ZooMu Cyber Infrastructure Working Group are to develop common collection management software language between existing databases using a data standard such as Darwin Core, to identify strategies for open zoo data to staff at museums and broader scientific community, but also respecting data sensitivity at zoos and discretion we need for certain species or certain kinds of data, and to develop a broad understanding of data safety and management. So what is ZooMu? ZooMu is an NSF-funded research coordination network. It is cross-institutional and designed to build networks. So the biodiversity collections in general, there's sort of two versions in a way. There's a living collections version, which is zoos and aquariums, and the preserved collections version, which is natural history museums and natural history collections. And we recognize that zoos and aquariums have data on an animal and their care from its birth. Usually they'll have information on that throughout its life until its death. And natural history collections are, I like to say, really good at showing you things that happened on the last day of an animal's life, it may be a little bit before. But then we also have data on all of its uses downstream from that. And you see the timeline goes to a little infinity. That may be a little optimistic. But the idea being that we in museums maintain data on the specimens and their usage ongoing as long as there's something there. Um, and I also point out to people when they ask me any of these kind of questions that the data, we curate the data long after the specimen is no longer usable. So if it's a tissue sample and it gets completely consumed, museums still maintain the data. The PIs and co-PIs are myself, Sheila Poo from Memphis Zoo, Stephen Whitfield from the Audubon Nature Institute, and Alex Shepak, a postdoc at Florida International University. Our advisory board and our steering committee include a great diversity of people with awesome backgrounds. We have museum people, we have researchers, zoo people, AZA representative, and all of our work is evaluated by an external evaluators group called Inform Evaluation. And they do a good job of keeping track of what our goals were and did we reach them. So plenty of surveys happening there. The timeline and how you got to where you are. We started the idea of ZooMu back in 2019 at a conference at Snowbird, Utah. There was a symposium on the expanding role of natural history collections. And notably absent from that was any talk on living collections. And so we got together and decided to do something about that. And we had two web-based workshops funded and run by iDigBio. We published a paper in Bioscience on building the network between zoos and museums. We held several symposia, including symposia at the joint meeting of ichthyologists and herpetologists, the Society for the Preservation of Natural History Collections, the Zoo Registrar Association, and the AZA. And then we got a grant from NSF to build networks. And if you look, we are probably pretty much right in the middle of that last chunk. We are in 2026. So you are here. All right. We like to say that zoos and aquariums and natural history collections are in two parallel universes, but they do a lot of the same things. So both have physical collections, including, in the case of zoos and aquariums, living animals, but also blood samples, histology slides, and things like that that veterinary services would have. And natural history collections have preserved specimens, tissue samples, histology slides, blood samples, etc. Both universes have computer databases they maintain to keep track of the collection. And both have people who do things, and a lot of energy spent to train people to work in zoos and aquariums or to train people to work in natural history collections. And one of our goals ultimately in the human capacity is to train people to do both. The Cyber Infrastructure Workshop was held at Duke Lemur Center about a year ago, a little more than a year ago. And as you read the caption with all the great people listed there, notice that there are people from software companies, zoo people, museum people, data research users. We had people from the two main zoo software companies, so Trax and Species 360 were both there. And we had people from Arctos and from Emu, Axial Emu. And it was probably the first time that those four software companies were in the same place at the same time. All right. At that workshop, we dealt with a lot of cyber-related issues. Among them, zoos and aquariums have a lot of sensitive data. The suggestion is that we look at how HIPAA compliant data are managed in human health, and it's a similar concept for zoos and zoos and aquariums. One of the problems, though, is that data are not interoperable with other biodiversity platforms, meaning that even within zoos, an animal moves from someone who uses Trax to somebody who uses Species 360, and the data may not transfer properly. And for certain, they wouldn't transfer to any of the data aggregators on the museum side. On museums, commonly we use Darwin Core as a metadata standard. There are other metadata standards in use as well. And we rely heavily on adhering to the FAIR data principles, which are findable, accessible, interoperable, and reusable. So the data need to be in a place people can find them and a place people can get to them. All right. In terms of accessibility, zoos and aquariums collection information is available. At least what is at the zoo can be found. If you think about all those photographs people take and put on Facebook about their visit to the zoo, that can be found. You can ask ChatGPT to make you a list of all the Komodo dragons in the United States, and it'll give you a pretty accurate list of who has Komodo dragons, even though it's not really looking at zoo inventory data. It's looking at web pages and maybe even Facebook. Species 360 allows requests for data from people outside Species 360, and it will be reviewed by external scientists and some internal review. Natural history collection had a major shift a while ago to make data accessible through data aggregators, and this was not a straightforward process. There were many people who took convincing, um, and it eventually came around to most people signing on. There are still museums that don't, so um, don't worry. Some people still don't share their data with aggregators. But like zoos, many things in the natural history collection, we can keep them isolated from the aggregators. We also have sensitive data. We have data on endangered species. We have data on extinct species. And so we can actually hide that sort of stuff. But the aggregators do a great job of collecting all this information and putting it into one place where people can find it. And on that, I will hand it over to Dave, and he will talk some more about that.
Thank you, Greg. Now that you've got a bit of history, I'm going to try to set the stage for some folks who come after me to give some examples and case studies of what we've been doing in this sphere. But before I do that, I want to clarify, kind of give you a sense of some of the players in the that we deal with, some of the acronyms and other terminology that you're going to hear, just to try to clarify how all of this comes together. I guess I'll tell you, Greg, next slide. There we go. Look at that. Almost like magic. Well, now it is entirely magic. There, we've literally just pulled a rabbit out of our hat. Perfect. So, one of the names or acronyms you're going to hear is GBIF, and that's the Global Biodiversity Information Facility. This is an organization I've worked with for 15, 16, 17 years now in various capacities. And GBIF is essentially the granddaddy of biodiversity information on Earth and its aggregation. They have a portal that you can search through, but they also provide other services to help people to clean, manage, and publish their data to that data portal, that data aggregator, and as well as others. I'm one of those people that helps with that sort of process. It is international in scope in a big way, both in terms of the people who are a part of GBIF as well as the people who use it. And one of the things that you would need to do if you wanted to be a part of this will be, or if you're considering the possibilities, you become a publisher to GBIF. And that is a free thing to do. It doesn't cost any money. There are no other obligations. And to do that, if you were curious, you would use the how-to drop-down menu in the upper left of their website and go to how to become a data publisher, where you can learn more about that. Of course, you can always contact almost any of us here on this webinar to find out more information about that. Greg? Thank you. To give you an idea of what I'm saying about sort of the largest clearinghouse, if you will, about occurrence data. So right now, at least as of a couple of days ago, there are more than 3 billion, almost 4 billion with a B, occurrence records. Occurrence records are essentially just a record of a thing in a particular place at a particular time. And that can be either, in this case, captive, or it can be wild. It can be observed, or it can be captured. It varies depending upon the institution and the purpose for the observation or capture. And all of that information, all of the associated data that comes with those sorts of observations or capture events goes along with it. Um, you can see that there are quite a few publishing institutions of which you might become the 2700th. How do you say that? That's a tongue twister right there. But that's entirely up to you. You can also see that there's a lot of effort now being put towards the serving of media files, images, and sound files, among other things. That's a growing source of data. Greg? Now, GBIF isn't the only portal or aggregator that's out there. For those of you that work in aquariums or other, or even in zoos, but certainly aquariums would be interested. There is OBIS, the Ocean Biogeographic Information System, which focuses on marine data and marine biodiversity. There are other aggregators out there. VertNet, which is the one that I maintain, is focused entirely on vertebrate data, even though I help to support people who have more than vertebrate data to get their data into other portals like GBIF or iDigBio. And you will hopefully in the coming year find that we have created our own portal specifically for data about zoos and aquariums. It won't be labeled or tagged as ZooMu necessarily, but we will certainly let the community know when that portal goes online. Correct? Uh, Another thing that you might hear, another name that you might hear is something called GR which is the Global Registry of Scientific Collections. This is essentially a directory of scientific collections, both the pinecone collection that your neighbor has under their mattress in their home, as well as scientific collections at institutions, government agencies, nonprofits, NGOs, etc. And this is a great first step to put your institution on the map, your information out of the world. It's free. It is administered by GBIF, and there are people like myself who are editors of various regions, like North America, for example, or South America and the Latin American Caribbean area. Um, all it takes is a simple how-to uh, search there on their menu bar, and you can learn very quickly how to add your own institution and list its collections. We also have some resources that we're developing that will help you with that to do that quickly and easily. But this is a great first step if you'd like to enter your institution into the world of biodiversity data sharing, and it's something that we recommend strongly. Greg? The last organization I'll mention is a group called TADWIG, which of course is not really a direct acronym for Biodiversity Information Standards. Forgive us for being obtuse as we are sometimes in the standards community. I'm the chair of TADWIG at the moment, and we maintain and develop data standards for the sharing of biodiversity. Go ahead, Greg. Uh, a couple of those standards that are particularly relevant. You've already heard Greg mention Darwin Core. Darwin Core is probably the primary mode of data sharing for biodiversity data globally. There is another one called the Latimer Core, which is similar, but works a little bit more about, tells us a bit more about collections and about those collections and what's in them. That's called Latimer Core, and that's what is used by GR Cycle. The Darwin core is used by GBIF and OBIS and VertNet and iDigBio and all of these other data portals. And there's a lot more to it. If you're ever interested in contributing to or would like to understand how standards get made or maintained, you can let me know. Greg? And there we go. Thank you. Uh, that's exactly the one that I want. Sorry, I failed to delete that other slide. Operator error happens. This is if you were having any questions about whether or not you wanted to share your data and what it does or what influence it may have where data comes from. This is essentially the chart that gives you the idea of the scope of a data sharing community. With GBIF being in the center there because they developed this slide. But you can see off on the left side all of the various sources of data, and on the right side, you can see all of the types of data and organizations who use that data that's available. And for those of you that are in zoos and aquariums, conservation is very important. Climate change, ecology, and evolution may be very important to you, and so all of this data is available in those for use for those particular purposes. So the question is, why would you want to do this? Well, other than the fact that we'll love you forever, if you do decide to share your data, first, obviously, the opportunity to contribute to biodiversity knowledge is important. To contribute directly to conservation and scientific exploration is important, as is the contribution to policymaking at local and global levels. Um, Joining the data sharing community also gives you your institution visibility and the opportunity for collaboration, especially between museums and zoos. Uh, Once you get into into GBIF and into GR Cycle, it gives you an opportunity to track your data usage and see who's using it and track citations when people publish papers and other things that utilize your data. Uh, as Greg mentioned, it's an opportunity to promote fair data sharing and fair data standards so that lots of people can use it. And also because zoos and aquariums are very underrepresented in this community and are much needed because your data will fill lots of gaps that researchers and other data users, educators could use to benefit their own work as well as your own colleagues. So I will leave it at that, and I will pass the lectern, such as it is, to Amanda Maza.
Hi, everybody. I'm Amanda Mazza. I am the registrar and the data manager at the Duke Lemur Center. And today I'm going to talk a little bit about how we started sharing our data through GBIF. Next. Just a little bit of background about the Lemur Center. We were founded in 1966. The Lemur Center houses the most diverse population of lemurs outside of Madagascar. And to date, we've housed over 4,500 individual lemurs representing over 40 different species. We're known for our non-invasive research and our conservation breeding for critically endangered lemurs. And besides our living colony of lemurs, the DLC also has an on-site biobank with historical and current lemur specimens ranging from tissue samples all the way up to cadavers, all of which are available to researchers by request. Besides the Lemur Center, we also have our Lemur Center Museum of Natural History, which houses over 35,000 fossils, an osteology collection that includes lemurs once housed at the Lemur Center, and a growing archive of digital scans. And these two pictures at the bottom are kind of representing the way that we store all of the data that we do collect at the Lemur Center and at the museum. And it's also kind of showing our long-term vision of connecting these two data siloed groups of information from the DLC and also from our museum. Next. So why did we publish in GBIF? So as Greg mentioned, we had our cyber infrastructure meeting in March. And one of our key outcomes from that meeting was looking at how we could leverage platforms like GBIF for a broader data integration and discoverability, which was two things that the Lemur Center has been very interested in doing and have been trying to figure out on our own how to do. So for us, we thought that GBIF might offer us a practical solution, and we were also willing to try to figure out how we can incorporate our data into GBIF, because again, our data has been siloed. Some of our living collection and our specimen collection data wasn't easily or readily available and tied together. And also our data is a little bit hard to discover externally. So for us, again, GBIF just offered sort of a good start for data publishing. And so in the past year, the DLC has published 4 data sets in GBIF, spanning from a metadata data set, a checklist, and a recurrence data set. So I'm going to go through 3 of the data sets that we've published so far. Next. So the first data set, so I'm also kind of starting from easiest to a little bit more complicated. So our first data set was probably the easiest to put together. It's our biobank metadata data set in GBIF. The image on the left side sort of shows what the landing page would look like for that. And essentially, this is just a descriptive write-up of what we have at the DLC Biobank. We don't actually have the list of the specimens yet. That is our end goal. But for right now, it's kind of a placeholder telling folks, hey, we're here. Here are the species that we have, the type of samples that we have, and if you're interested in getting these samples, who you can reach out to at the Lemur Center to request this type of information. So it was a really easy thing for us to put together. We had some of this already pre-written. It took maybe about an hour to throw together, and it was a quick, easy way to get our name out into the land of GBIF. So then we decided to do something a little bit more complicated, but it was not that difficult once you understand what what the GBIF standards are. And we decided to publish a species checklist. So this is essentially just an inventory of all the taxa that we've held at the DLC, both historically and currently. And it was simply generated by using our collection management system, which is through Species 360, and it's known as ZIMS. I generated a report in ZIMS to just show all of the species that we've ever had. I exported that into Excel. The hardest part of this whole process was just cleaning the data set that I got from ZIMS in order for it to meet the standards of GBIF. And then I was able to publish our checklist, which is the final product that you see in this data process flow. Overall, it took roughly maybe a few hours to kind of put together, but that really also just depends on your collection and how extensive your animal list could be. And it's also totally up to you as a zoological institution on what you want to put out there. If you don't want to put what you currently had, that's okay. You can just say what you're currently exhibiting at your zoological institution. This is all really up to your institution and what you feel comfortable putting out there. But this also is just a valuable list that we never really had other than just on our website. So it just gives us a little bit more discoverability out into the research community. And then the other data set, the last one that I just want to talk about is our osteology data set. It is essentially the most data rich that we have out there so far. This is an occurrence data set, which David mentioned previously. And what we did here, so again, an occurrence data set is just like, here's a specimen at this place at this time. But because our osteology collection does house lemurs that lived at the Lemur Center, we decided to make this a little bit more robust than just here's the osteology collection at the Lemur Center during this time. What we decided to do also was include information from this animal when they lived at the Lemur Center. So we linked their house name, their DLC ID, their GAN, which is their global animal number that we have from our collection management system, ZIMS. And we also used their body weight data, also pulled from ZIMS. And we connected all of that to a digital scan of our skeleton, which we're storing in Morphosource. And Morphosource is a publicly accessible 3D data repository where most of our scans are currently being held. So ultimately, I took all of these different siloed data sets and combine them in order for us to import this information into GBIF to get this wonderful display of all of this connected data. And so this did take, I will say, some effort between me and one other person at our museum. But the combined effort was completely worth it in my mind. And so once we did this and we understood the process, we then published our Egyptian fossil collection, which you can also find on GBIF. And we are now going to move forward. We're publishing two more data sets for our most critically endangered species that we hold at the Lemur Center. Next. The other thing that we really liked about GBIF 2, we didn't have the best method of showing how our data is used. We would collect that information at the Lemur Center and at the museum, but this actually helps us a lot more through the GBIF activity reports. As Dave mentioned before, you can see when someone cites one of your data sets, but it also shows the activity of your data sets. And so the Lemur Center and our museum have really only been on GBIF for less than a year. And as you can see in our activity report, two of our data sets have been pulled multiple times, almost every month since we started to publish. And that's just a kind of a collective report on the left side. And on the right-hand side, you can see more detail about just the fossil, our Egyptian fossil collection, and how many times that's been downloaded in an event in a big GBIF pool. So for us, this is also really helpful because we've used some of this already for reporting purposes. We've used this also in some upcoming grants that we're writing. And this also is really good for me to just show to some upper management and justify like why this is important for us to start publishing our data out to GBIF. So it's been helpful in that manner too, just to explain why we're putting all of this work and effort into putting our data out into GBIF. Next. My last slide is really just some key takeaways that I've learned by doing this process. Number one is just start small. Do the easiest thing if you're interested and you want your zoological institution or your aquarium just to get their name out there at first. That metadata data set is very helpful and very easy to put together. Again, if your zoological institution or aquarium has a collection management system like Trax or ZIMS, you can easily pull a taxon list down. Simply clean that to meet the data standards for GBIF and import in. It's a very minimal amount of work for a pretty big impact, especially if your data has never really been out into the research community. Use what you have. Like I said, most of what I have put into GBIF so far is just out of ZIMS. We have other databases where we hold more data on these animals. So in the future, I will start to integrate that data as well and build over time. This is just what we've done in 1 year. Like I said, we have many more data sets to publish. So there's no time crunch to get your data out there. So feel free to take your time, use your resources, and finally, scale it to your institution. The DLC is a very small institution. I am the data department. We have one other person in our museum that also helps me. And with the two of us together, we've been able to publish these 4 data sets in a pretty good timely manner. And it really didn't take that much extra effort to put this together. But I think for us, the impact so far has been a lot bigger than we anticipated. And we're just even more excited and encouraged to get our data out there, combined finally to sort of create this extended specimen network that we've been dreaming of at the Lemur Center. So it's been a great experience for us. And I hope it helps other institutions take some of the fear away from putting your data out there, but also just, where to start and that you just need a little bit to go a long way. Thank you.
Thanks, Amanda. Well, hello. My name is Cameron. I'm the Oh, so I keep forgetting my title. I am the assistant collection manager of vertebrate zoology at the Denver Museum of Nature and Science. And so this is kind of taking a look at how two separate institutions can kind of do a similar thing to what Amanda and David have mentioned. And so with this, this is kind of like how we have built up collaboration through two places that have been founded for a long time and have worked together in some capacity, but now we're trying to integrate even more together with this. Next slide. So I'll talk a little bit about the Denver Zoo Conservation Alliance. And so it was founded in 1896 in City Park here in Denver when a black bear cub was given as a gift to the mayor and so he was like, I don't have a place to kind of store a bear. And so the zoo was brought up as a initiative to house a bear. And then they were like, oh, we can actually build on this as well. And so a lot of regional animals were brought in to the city to start the zoo. And the initial focus again was acquiring native animals, and so the zoo had a focus on that, and it's one of the first zoos to use a naturalistic zoo enclosure. So if you hear about Bear Mountain, that was at the Denver Zoo. Next slide. And then right next door is the Denver Museum of Nature and Science, founded in 1908, where the zoology collection was really the first thing that was cataloged and described and brought into the museum, collected by Edwin Carter as late as 1968. And so it was kind of the stepping stone for creating a museum institution in Denver for this purpose of, maintaining and preserving natural history, but also exhibiting it in different ways. And so over the years, there has been expeditions all over the world done by the leadership here at the museum to bring about more animal collections and research capabilities. Next slide. So really, we can think about these two institutions working together, and the easy thing to think about is just transferring an animal from one institution to another. The earliest recorded case of this is in 1903 when a fallow deer and a moose that passed away at the zoo was transferred to the museum to be used in their research collection. And so over time, communication was set up between veterinary staff, but now it's kind of shifted to the registrar at the Denver Zoo to then work with the zoology staff here at the museum to help keep that transfer agreement and kind of connection or the bridge together. So animals that are transferred to DMNS are processed into the research collection, and the data is published into the collection management system Arctos, which is a database portal similar to Species 360 and Tracks that is mostly used for museum material, for natural history museum material and collection of data. So that is a system that is available that also feeds into GBIF and otherwise. And so those systems are what museums use primarily, or what my museum uses primarily in the zoology. Next slide. So when we think about data sharing, we're talking about digitization of the metadata inside and outside of the museum, in the sense that we have identifiers, we have attributes, we have measurements, and all these kind of data that are connected from the zoo side when they're collected during life. And then once the animal is transferred over, we have a new set of data that is made when the preparator actually goes through and does similar measurements. I would say, taking tail length, things like that, weights, we do that. And so we kind of are creating two kind of subsets or two kind of data sets from that. And so the point of this is to kind of bring them together. This link that was posted is the project page on, you have to have a login for it, but it kind of shows all the different animals that are transferred, that have been transferred to the museum from the zoo and have been digitized. And so there actually are more things that are in process. We have things that are in our backlog that are going to be processed as soon as we can. But then this kind of lists out some of those. And inside the project, you can actually see citations. You can see all the catalog records. You can see the projects and the agents that have been associated with this specific project. And so that's a way that Arctos has been trying to publish and to be able to share that kind of information out. And so to talk about the kind of process of that, I was going to go to the next slide. This is kind of the step-by-step process that we do when we bring in animals from the zoo to to the museum. So if an animal is notified, or if we are notified that an animal has died or passed away, we will be offered the animal afterwards, and then that animal will be picked up and brought back over to the zoo, to the museum, where we will then process it. Larger animals get processed the same day, just to save space in our freezer. But then if we can take our time, we will put it into the freezer for storage. And then we will prepare that animal into different ways. If you're familiar with some methods or preparation procedures at museums, we can make a study skin or a skin clip. A skeleton is prepared as well. Tissue samples are taken, and then standard measurements as well. And so once that is done and the prepped animal comes down to the collections where I work in, then that information will be uploaded into Arctos. That will be associated with the catalog record, and we have some attributes that are given to us from the zoo to the museum that we can also use into that system. So it includes identifiers that are made at the zoo, the origin or the sex or like other things like that that we have gotten information from the zoo that we can publish in. So now, after that, through Arctos and GBIF and these kind of data aggregators, we can have researchers pull information from these sources and then request it if they ever have any questions or further research. So we have plenty of requests that come for all of our specimens. We have about 25,000 mammal specimens. We have about 60,000 bird specimens, and some of those portion of them are from the zoo. So it's kind of cool to see the data kind of live in that space and then be used in different types of research projects. Next slide, please. So in the future, we have been talking more about how to better integrate these two systems. And so our plan still remains the same, of that animals are picked up at the zoo, brought over to the museum for preparation, and then any information that we have or gather during that process will be uploaded into Arctos. But we want to have this cycle back into the zoo where they can also utilize some of that information. So if we have given permission to the zoo or have shown them how to get into Arctos and then give them permissions to actually access some of that data that is directly impacted by their own collections, their living collections. And so they can see all the use cases, all the publications, all the citations that are housed in Arctos, and then they can bring it back to the zoo and use it into and integrate it into their systems. And hopefully one day we can find a way to kind of make that easier just by having those two systems. Denver Zoo uses tracks, and so having tracks and Arctos just talk directly together to have that be easier. And so that will be a continuing discussion as we go forward, and things are already in place to kind of make that easier. Next slide, please. So the future of our collaboration is that we want to improve the exchange of animal data guided by research and availability. We have seen that there are different communities that reach out to either one of us instead of both of us to kind of find that information out. So this is a way that we can actually bring both pools together, and then using different ways of refining our digitization efforts. So, there are CT scans or data like that are taken at the zoo, and we also do that same thing at the museum. So there might be a way to kind of take out the middleman and actually just have a system for both of us in place. And then the consideration of implementation of biobanking. So we have at the museum a cryo vault facility or a cryogenic storage facility that stores our tissue samples, and we know that the zoo does as well. So we're trying to figure out a way of what can we do to help both of our institutions kind of continue with that project. Next slide, please. And so we have some things in the works. We are working on a kind of a guideline and manual because it's going to be kind of important that we have a kind of a case study on how both of these work together. And then we have, of course, added things to Arctos operationally. And so we're going to try to get both institutions, of course, the museum is in GBIF and GR-Cycol and Morphosaurus, and we're trying to get the zoo in as well as kind of collaborators in that sense. And then we're actually working on some signage and publications at the museum or at the museum and the zoo, because I think it's important to talk about the outreach. And we have open line of communication with guests here that, a source of animals that we do have for our zoology collection comes from the zoo. And then they always ask, like, that makes sense. And like, how does that work? And so we always try to answer as many questions as we can about it. And I know the zoo is doing the same thing when they talk about an animal that has passed and being transferred over to the museum. And so we're trying to work on ways to better share that message out to the community around. Okay, next slide, please. Oh, I think that's actually it. Okay, yeah. So next slide is next steps.
And that concludes our presentation portion of this webinar. I'd like to thank our presenters. That's Greg, David, Amanda, and Cameron. And also would like to thank NSF for funding this initiative. If you have any questions, please feel free to raise your digital hand. I think there's one up already, or type your questions in the chat. And I wanted to end with good news that while this is our first webinar, it would not be our last. So as we continue to develop the cyber infrastructure for data transfer and integration from zoo databases to museum databases to data aggregators, which includes a new ZooMu portal, we will be holding future topical workshops, including things like how do I put my holdings into GR Cycle, or what is GR Cycle? And then we will also be continuing to present at conferences, including ZRA, and also holding regional in-person meetings. So please, tell us what you want to know and learn about and help us shape our next steps. You will see a QR code on the screen. Please take a minute to give us feedback. And also you're welcome to contact us, visit our website, watch our YouTube channel, and email us for any questions or just to say hello and tell us what you think. So with that, I'll turn to question and answer portion.
I just let Emily talk, so if you've got a question, go ahead and discuss it.
I was going to answer Stephanie's question. If you get an animal soon after death and necropsy, what are the precautions as far as biosecurity and zoonosis possibilities? So when we pick up animals from the zoo, they have been already necropsied, and so they have gone through the cases of whether or not it is safe or deemed unfit for transfer. So we're not picking up every animal. We have plenty of cases where if it is unsafe for anybody to handle, we will not receive that animal. But then in cases that it seems fine, we will of course use the same precautions that they use when they do necropsies. So that is using PPE, gloves, and all that. And we have the same level of safety at the preparation or the prepared prep lab, I guess I would say. And so we kind of follow the same guidelines for that. So it is easy for us to transfer animals and make sure that everybody that works on the animals are kept safe. Hopefully that answers your question.
Were there any other questions?
There was a question about full necropsies in the Q&A. Maybe there could be a better response or a more full response from especially the zoo side. Can you see it? It's under the answered. I did respond since it was a topic of previous ZooMu workshops, but I think it could be expanded on.
Yeah, I might say this one again since it's from the Denver Zoo. So yeah, sometimes we get an animal that has been fully necropsied, and so that might change what we take or what we prepare in that sense. But in the we rarely get something that is fully intact. So they do have a pretty in-depth system of doing their necropsies. And so we are happy to work with them on kind of what we're looking for in terms of the best case for making a study skin, a prepped material. And so we have open discussions about that going on and going forward.
I know there's also a movement to do necropsies in a more cosmetic fashion, where the information that the zoo vets need to get can still be achieved, but the specimen is as intact as possible. And that does help. And we've had birds come in where we end up keeping a tissue sample in a wing. And there's still some data to be gotten from those wings and those tissue samples. Nancy's asking a question in chat.
I can answer that just from the side of the Lemur Center. So we don't have any worries.
Oh, first of all, please repeat the question because I
Oh,
don't think all
sure.
the participants can see it. Sorry. Go ahead.
Thanks, Michelle. So the question was, has there been any worries on having the data be used for bad, i.e., find number of animals that died at a facility? So one thing with the Lemur Center is we are a very transparent institution. Uh, if you go to our website, we do have information and data sets out there where we have had folks question what happened to an animal. I saw, 2 months ago, this animal was on your list living in a natural habitat enclosure, and now I don't see the animal listed any longer. What happened to it? And we are very forthcoming with information. We do let people know when they have these questions that unfortunately, animals either move to another institution, and that's why you might not see it anymore, or the animal has passed away. And we have yet to have really any negative feedback from having our information on our website or even what we have in GBIF so far. I think because we do come right out and answer these questions, that some folks who may have started out with a negative thought have then since changed their thought by just having some open discussion with us. So while we've always had that sort of open communication with the community, we haven't really had any negative feedback or pushback or any bad intentions. But we also believe at the Lemur Center, I think that there is a bigger impact putting your data out there rather than hiding it for the fear that one person could, misrepresent, misuse, or misinterpret your data. So we have, really hadn't had any issue with that. And I don't think in the foreseeable future we will ever not publish our data because of that issue. But again, that's it's up to your institution and the comfort level that you have.
I think that builds into the question that Ariel Woodward also posted in the chat about data that might need to be hidden, masked, or obscured for potential sensitivity issues like law enforcement data. I think it would be up to the institution to be able to determine what information they can put out there for open access. And of course, there's also a situation where you might need to ask permission if you're putting data about loaned animals out there. You might need permission from the actual owners. Not everything can be open access. Not everything should be open access, and so it's a matter of being able to configure your data and what you then integrate into the data sharing platforms.
Yeah, I can also follow up. This is an ongoing museum concern as well. So we regularly encumber, hide, make private locality information, species identifications, things that may have some species of conservation concern, for instance, and so those are not shared with the public, and they're not shared with GBIF, obviously, because that would be all public data. So there is a layer of filtering that happens even with the stuff that we share, and we can continue any concerns that a zoo may have with the specimen they're donating it, maybe only make it available to researchers, but not the general public. So there's lots of nuances, I think, to the data sharing. But I think the most important thing is that we have mechanisms and protocols for making these decisions and who has access to them. I'm sorry, I just noticed that Dave has his hand up. Sorry, Dave.
You don't be sorry. I just raised my hand. That's all. I just wanted to address both of those. First, in the 25 years that GBIF has been publishing data, I don't know of any instances where that data has been taken and used for nefarious purposes, either against an institution or an individual. Certainly anything is possible, but the vast majority of the people that are utilizing the data from those data portals are using it for their research, for conservation, for decision making. It goes into the Kunming protocols, it goes into the old Aichi targets, it goes into all of that. I don't know of anybody who's ever had to pull data because of somebody who's trying to make a point or score political points in any particular way. And further, just to support what Michelle said, yeah, there are processes and protocols that we have for doing things like for locations. If you've got like the Smithsonian Zoo has both animals at its facility, but it also has a whole bunch of research and photographs of from expeditions that they've done in the field. We can always either redact or fuzzy, if you will, location data to protect sensitive species and sensitive habitats. But ultimately, at the end of the day, if you have data that you're concerned about for one reason or another, you're not obliged to publish it at all. That is, the decision of what gets published to data aggregators is entirely up to you and your institution. And so you have that control. And should anything escape your view and we need to change things, that's a relatively quick process to take care of through GBIF and other portals. It's not something that takes months and months to change. It can happen pretty much overnight if need be. So if you had questions about those sorts of things or about specific data, I'd be happy to answer those after the webinar's done.
I wanted to add also to Stephanie Earhart's posted a question about can tissue samples that are sent to outside lab for histopathology, could what is left over for that be useful to a museum? And Cameron replied that yes, it could be very useful and valuable to museums, but also the data that's generated from the lab results from that histopathology. One of the ways that could be shared with museum is the data. And that is if you're cataloging your histology slides and the data and diagnosis from it, that could add to as a reference data for part and parcel of the source animal that the museum can use if they have other parts from that same individual animal. So there's a lot of ways that you can go about it. You can share, in fact, the images, but you don't even have to have a preview image. Just the metadata that is coming from your database could be transferred into a data aggregator for other researchers, both in museums and outside other scientific institutions, to be able to find that. And then they can contact your institution to ask for the images. Or you can use that to share into larger repositories for veterinary medicine and those types of initiatives that I see popping up. There's a lot of reference materials out there that you can use your data to share outside.
Yeah, tissues are rare. They're hard to come by for a lot of people. So beyond museums, just the research community, the conservation community having the sorts of things can be the difference when you're not.
All right. Looks like we've come to an end. Or are there any additional questions? All right. Well, thank you all for attending our webinar. And like I said, it has been recorded, and we will send a link to that once it's posted to everyone who registered, not just the ones who attended. But all right. So thank you very much.